discussed most of what we wanted to say about the qualitative aspects of the tannins, but we hadn't said enough yet about the anthocyanins. So picking up again near the bottom of page 2, you notice that if we take the data largely from Professor Webb and Kepner and their students, taking 117 varieties of Vitis vinifera, grape varieties, that did have acyl pigments, acylated pigments, the average content of the different 3 glucosides are as shown, with 3% delphinidin derivatives, 5% petunidin, 41% malvidin, and because on the paper chromatogram cyanidin runs, cyanidin 3 glucoside runs in the same position, the cyanidin would be included here, although the amount appears to be small from other studies. About 10% pionidin, and then the caffeic acid acylated pigments as a group, which would include apparently all the previous ones, but with a caffeic acid group acylated to it, would be around 14%, and 26% of paracumaric acid acylated pigments. So you see there's a good chunk of the pigments that are acylated, and because they move then as a family, the acyl group has fairly controlling as to how they migrate on a paper chromatogram, then they haven't really been separated. We don't know whether the anthocyanidin ratios stay the same, but we would assume they would, since the acylation is a late application of additional unit. Chances are the ratio of anthocyanidins would stay about the same. If that's true, you see that the grape is uncommonly, the European grape is uncommonly high in the methylated pigments. Cyanidin being small enough to hide behind the malvidin, and then all the others except delphinidin are methylated at least once, and delphinidin is not large in the Vitis vinifera group. This has some significance in that this no doubt contributes to stability of the wine pigment. And we don't need to go into this very deeply, but if this is at this point, but if this is the B ring and you have a methyl group there for pionidin and another one here for malvidin, this would certainly tend to prevent oxidation. As illustrated, if we took petunidin, which has this structure, then it would be very easy to oxidize that to the orthoquinone form relative to the ones with the methyl group in the available positions. So that being so, petunidin would be a good deal more oxidizable than either pionidin or malvidin, and of course delphinidin with the pyrogallol type ring would be the most easily oxidized of the group. Cyanidin also relatively easy. So at least in terms of relationship to oxidation, it's rather good that the main pigments of red vinifera wine would be the highly methylated forms. We have said nothing here about the acetate acylations, and the reason being that these were discovered after the ratios here were worked out, and as I said before, the acetate groups seem to come off so readily that they're probably unimportant in any wine reactions, or for that matter, difficult to even show their presence in grapes. So while it's very interesting and probably has some importance in terms of relative solubility and other factors such as that in the plant cells themselves, then it probably has, at least we don't know of any significance once the pigment is separated from the grape skins, as in red wine. Now, if we take a look at 8 varieties that lacked acyl pigments, the ratio of the aglycones, the anthocyanidins, was apparently somewhat similar, but it does, if anything, emphasize the increase or the high level of malvidin. So not only is the suggestion present that the ratio stays something similar to the to the distribution of the aglycones with acylation, the suggestion is, if these 8 varieties are typical for other reasons, that in fact the malvidin would tend to be more readily acylated, and the percentage is in fact higher than would be indicated by the previous information. That's perhaps reasonable, since methoxyl addition is a late reaction and acylation is a late reaction. then if you're emphasizing late reactions, they might tend to go hand in hand. In any case, this is somewhat different from the Vitis labrusca types. Note that the Vitis labrusca types were much higher in delphinidin content and proportionally considerably lower in malvidin, and also more cyanidin, so that the emphasis in the conquered types would be toward the less methylated and the more hydroxyl type of pigments. This has considerable meaning in that, if you stop to think about it, you don't see very many blue vinifera grapes. In fact, I can't think of, there are, I believe, none that have, in fact, even among labrusca, there are none that have a dilute blue color. There's no such thing as a pale blue grape as there are pale pink grapes. So that, and most of the pale colored grapes that we know are, in fact, vinifera. So that you get a conquered grape that's a rather blue color, almost as blue as the outside of this plastic here, but that's partly due to the bloom, which tends, the waxy coating, which tends to dilute the color visually, but nevertheless it is blue, whereas Pinot Noir or others tend to be called black, as their name implies, and are not blue. They may be purplish, but they tend to look more red, and if you take the juice away from them, there is more of a red color in the typical vinifera than there is in the typical conquered type juice. In fact, in conquered juice, they have some trouble for slightly different reasons, but at least it illustrates the point, with an actual blue pigment that precipitates out in the tank along with the tartrates and so forth, so that you can isolate blue pigments from conquereds, and it's never happened to my knowledge with the vinifera. Well, part of this, at least, is explained by these relative percentages then, because delphinidin is blue. If you have trouble remembering that, maybe you know the flower delphinium is certainly blue. So the delphinidin derivatives tend to be blue, whereas the methoxylation tends to convert them back toward red. And we'll have a little more to say about this when we talk about spectral color, but that's typical. So that the pelargonidin, which doesn't occur in grapes, tends to be a salmonish buff pink. Cyanidin tends to be red, delphinidin tends to be blue. And then if you methylate either cyanidin to give peonidin or delphinidin to give the malvidin or petunidin, you get a shift back toward the red. Well, at least then it's possible to say that the colors are somewhat reflective of the mixture of anthocyanins that are present in the particular grape. Yes?
What's the case with French hybrids as far as...
Well, the only clear-cut answer we can give you to this, well, there are two things we can say. The French hybrids tend to be, we tend to think of them as labrusca crosses. In fact, most of them are not labrusca crosses, or if so, it's labrusca cross with something else too. And so they tend to have extremely high color levels. I suppose because they were bred by Frenchmen and who'd been plagued by low color, one of the things they looked for in selection was high color. So it tends to be produced in the wild type grapes anyhow, particularly rupestris, which very often breeds in the teinturier character, the red juice, red pulp, red internal juice character. So you have very high colors. And as a consequence of the very high colors, you do have trouble noticing whether its skin becomes black in the berry. So you generally don't have anything that looks blue in the so-called French hybrids. And by the same token, once the color is out into solution, then even in conquered, it doesn't look so blue, it looks more red. So in wine, I know of no particular tendency to have a bluish color, although I think there is a small tendency in that direction. The main tendency that's easy to observe is a much higher color than we would be used to in many grapes, as high as our highest in many cases. And it's significant that Salvador is we don't think of it as a French hybrid, but it's a hybrid. It's a teinturier with very high color. Ruby Red and Royalty have rupestris parentage with very high color. And Alicante Boucher is subject to some debate. I haven't seen a clear-cut statement in the literature. They believe that at least the best information I have at the moment, they think that there was or is a teinturier grape, the grape itself called teinturier, which means dyer in French. This grape apparently is pure vinifera, they think, And then Alicante Ganzin was clearly a hybrid, so whether Alicante Boucher, which was another in the series, how much vinifera and how much non-vinifera it has is open to question. Anyhow, it's a pretty general statement that you can make that if you had a red juice grape, the odds are very good that it has some American, probably rupestris parentage in it. And since you've asked the question, I might as well come to the other point that I was going to make. The 3,5-diglucosides are present along with the 3 monoglucosides in the labruscas and most other American species, so that you have both forms, the 3 monoglucoside and the 3,5-diglucoside, whereas in vinifera you have only the 3 monoglucoside. Now this has been made use of in way I suppose you have heard about. For instance, it's illegal to grow in Germany and parts of France the crosses, the direct producers and the so-called French hybrids. The reason it's illegal, or at least the only logic, well, there are 2 main reasons. One is they don't want unfair competition. In other words, if you're growing the so-called noble grapes, the true vinifera grapes, and therefore have to go to the expense of grafting and other things, then they don't think it's fair, and the yield's probably low compared to the possibilities in the hybrids. Then they don't think it's fair competition to have somebody next door growing a cheap grape and selling his wine as Appellation Control A or whatever high-priced wine. So that's the main reason for the legislation against such varieties. But there is another interesting reason for it, which apparently is the controlling reason in Germany, and that is that you may know that phylloxera in some parts of the world, where it's native in the U.S. and in much of Europe where the humidity is reasonably high in the summer, has a flying form. There's a very complicated life cycle that I don't pretend to understand fully, or remember fully at least. I've read it, but in any case, it has several forms, and it can reproduce and maintain itself as the underground root louse type, which only navigates by crawling or being carried by man. But the flying form, of course, could spread much more rapidly and over a wider area. Well, if the flying form attacks a vinifera leaf, it doesn't seem to do much to the leaf. In fact, it may make a necrotic spot, but not much happen. But if it attacks a resistant vine, the resistant vine makes a gall. And then from this gall will emerge a different stage of the insect and fall on the ground and repeat the cycle. I'm not sure this is on sound entomological grounds, but the inference is, at least, that the types that don't make galls then would suppress the flying form, because the galls wouldn't form and presumably the insect wouldn't multiply as rapidly. Since the insect has bypass mechanisms that will enable the flying form to still propagate, I'm not, at least as I understand it, I'm not quite sure this is valid. But anyway, the idea is then if you prevent the propagation of the type that forms galls, the American species, then you would slow down the spread of any flying form of phylloxera, and for that reason it's prohibited. Interestingly enough, growing such grapes is prohibited in Germany, and yet they need rootstocks, obviously, which would fit the definition of gall-forming types. So they have an arrangement with, I wonder how much they made clear to the Italians what they were doing, but anyway, they have arrangement to grow the rootstocks in Italy and ship them to Germany as cuttings, so that, and pre-graft, bench graft, and that kind of thing, so that you aren't growing the prohibited vines in Germany, but let the Italians take the brunt. And I guess that's fair too, because there are, of course, direct producers in Italy already, so you're not adding to the load, I guess. In any case, I wonder what happens if a rootstock buds from the base, or if you freeze back. I suppose they watch this very carefully, but clearly it's the kind of prohibition that can never be 100% maintained. Well, the 3,5-diglucoside occurs then with the 3-mono in the typical American species, and this, as we've already said, is dominant. A dominant character. So if you made a one-generation cross with the vinifera, it always carries the 3,5-diglucoside. Now, of course, if you segregate those seedlings, and particularly if you back cross, then you can find crosses that don't have the 3,5-diglucoside character. But up to now, it's been reasonably possible, because the 3,5-diglucoside character seems to be more than a single gene, even though it's a dominant character, so that it does carry more than one back crossing generally. And it is possible to analyze the wine for a 3,5-diglucoside content and then say, well, this was made from direct producers if you find it, or no, it wasn't if you don't find it, and therefore such wines can be successfully prohibited from importation into Germany and so forth. Since they won't let their growers grow them, then they won't let such wine be imported either, for two reasons. One, they couldn't prove their growers were honest if such wine was blended in, and two, again, it's considered unfair competition. Now, I'm not quite sure what the European Common Market has done to this, since theoretically now we have an open path for Italian wine into Germany. I suppose if it's labeled as Italian wine, it may be permitted. I'm not certain about that. But it's certainly still not permitted to be blended with wine that's called German wine. Question?
I understand the Germans managed to stop it.
To which? Stop?
They stopped the importation.
I see. In spite of the common market. Okay, fine. I didn't know what the situation was.
Specifications on their Food and Drug Act that make it impossible to import things they don't want.
Okay, fine. Of course, this is a weak crutch at best, because in the white grapes there is no way you can, at the present, even in spite of looking for it, no way that you can tell the white grapes, since they lack the anthocyanins anyway, so they can't really detect hybrid white wines, and vice versa. It is going to be clearly true before very long. If someone wanted to go to the trouble of circumventing the law, it's going to be possible to select and breed crossbreds that lack the 5-glucoside form, and therefore would defeat the law. This has some bearing on stability of pigment. Interestingly enough, the 3,5-diglucoside seems to be less stable than the 3-monoglucoside. And there's been work done by Hrasdin at Cornell and others at Cornell on this point. It also affects the color a little bit, but not color mainly related to stability, and so it has some other possible reasons for interest. While we're on the point, it's interesting that the rotundifolia, Vitis rotundifolia, seems to be exclusively 3,5-diglucoside. It lacks the 3-mono, so if you wanted to go, and this gives an idea of the taxonomic relationships of some of these things, if you wanted to, if there happened to be a crossbred and you were trying to find out was it more similar to the rotundifolia parent, you could use that as a criterion. And there are Asian species that seem to be exclusively the 3,5-diglucoside as well. While we're on that point, we might as well bring up another somewhat similarly related subject, and that is the lack of the gene for acylation. The acylated pigments are absent from Pinot Noir and its close relatives. Gamay Beaujolais, I believe Gamay, and a few others that are rather closely related. And this is the reason for the group of the 8 varieties lacking acylation. So that the Pinot Noir group, if you wanted to tell whether a Burgundy was authentic, and if you could get it young enough so that there was still a good deal of the acyl pigment should have been present, if you find no acyl pigment, you could say it probably is pure Pinot Noir. If you find acyl pigment, you could say it certainly is not pure Pinot Noir. No practical use, to my knowledge, has been made of this, partly because the acyl groups don't stay on too long in wine, and therefore it's not too easy to check this out. But if you're buying grapes, for instance, it might be used as a technique to be sure that you weren't being fooled. This is somewhat interesting in a breeding sense. There are selections of Petite Syrah that lack the ability for acylation as well, even though they're clearly not related to Pinot Noir. And you can study the particular clones and sometimes their interrelationship by following that sort of thing. I don't know, again, of any practical use has been made of this, but at least Professor Olmo does have some Petit Syrah selections that lack acylation ability. Now, the anthocyanidins, the aglycones after the sugar is removed, do not occur in nature in any case known, as we've already mentioned, except possibly that flowering peach that we discussed. But in processed products, they might occur. The glycoside might hydrolyze off and leave the aglycone behind. Well, the evidence is that either this does not happen, or and more probably the anthocyanidins are too unstable to survive for any appreciable time, so that you cannot find, no one has been able to find any significant quantity of anthocyanidin in aged wine or any other product of this kind. On the other hand, the anthocyanins do form polymeric derivatives combining with other phenols, particularly the tannins, to make a red tannin, so that polymeric anthocyanins or red tannins, depending on what you want to call them, do occur in aged wine. And in those cases, it may be that the sugar may get hydrolyzed off and the molecule may be still stable enough. That's not been well studied, although it has been shown that there is still glucoside sugar within the so-called red tannin, so that apparently mostly the sugar stays attached. These polymeric tannins have been reported by Summers of Australia to occur in grapes, but we believe that's incorrect, and I think he at least has toned down his statements in that regard, so I kind of think he's convinced too now. If you run a two-dimensional chromatogram, or for that matter a one-dimensional chromatogram with fresh, well, if you run it with extract of grape skins, you'll get the anthocyanins wherever they happen to migrate. And the origin spot where you began, it may have some material which would be polymeric, and it's probably, at least it sticks tight in both solvents. One measure, one estimation of its polymeric condition. And it is red, or at least a degraded red, a brownish red, that sort of thing. If you get your preparation fresher and fresher and fresher, you can produce it with no origin spot. If you let it go to pot, you may get a lot of origin spot, and it doesn't take very much before you get nothing but origin spot. That being so, then we believe that in the grape, they don't occur as the red tannins. And we think that Summers' finding that they did was based on partial degradation of the pigment. Now, that may be unfair for other reasons. We feel quite sure of this in terms of the kind of polymer that's present in red wine, but it is true that the pigment within the grape skin may be a complex of some sort, different from that usually found in aged wine, which is sort of polymeric. And this has been more studied with flower color, and there are complex flower colors that include even pectin-like units that seem to be certainly associated, maybe bonded to the pigment, and co-pigments of flavonols and other flavonoids that are related to, cannot be easily separated from the complex, and for that matter metal ions like aluminum and so on too, and we'll say a little more about that some other time. But in any case, by and large, when we isolate, if we're making wine, if we make the wine as fresh as possible and look at the pigments, they're non-complexed in vinifera wine, anthocyanidin, 3 glucosides, and then as the wine ages, there will be complexes forming with other phenolic constituents in the wine, and you'll get red tannin-like polymers. Okay, I think that covers the points I wanted to make. Well, no, there is one other point that I wanted to make about anthocyanins while we're on the subject. I said you didn't usually find, we know of no dilute blue-colored grape, but there are dilute red-colored grapes, flame Tokay, depending on where it's grown, maybe Grenache and so on, are actually pink grapes. Mission is sort of a grayish color, and gray Riesling and Gewürztraminer are pinkish or grayish looking berries. Certainly we do have a dilute color, and it's kind of interesting what seems to happen. In the best studied examples at least, and Min Akiyoshi's master thesis was one of the best on flame Tokay, it showed that it was mainly the cyanidin 3-glucoside that was present in the dilute colored, at least flame tokay, and the others that have been studied. What seems to happen is that you lose then, the reason they're pink, they've lost the ability to produce the fully methoxylated types of compounds, so that you get cyanidin instead of the delphinidin and malvidin analogs that would involve further production. So just as we mentioned last time, if you go from a natural wild red grape to a white grape, you apparently lose the ability to make the pigments without producing an overflow into other phenolic compounds. And in this case, the same seems to be true. You lose the ability to make the methylated derivatives and therefore get a dilute red color or a pink grape instead of the usual fully methoxylated types. Yes?
I know from working, floating around like last year, some of the last vintage, things coming into the winery last vintage, especially on the Flora diversity and the Gray Riesling, they're coming in almost black.
Yeah. Well, there are other environmental reasons for difference of that type. You know from other classes that anthocyanin level is better higher if the weather's cooler, and we'll talk about what we know about some of those things when we get into the viticultural aspects, but that's one kind of reason. And what usually happens there is, again, if we had the two-dimensional paper chromatogram, and if some pattern is like so, well, let's exaggerate it. So we say we had this kind of a situation where this is little, and this is bigger, and this is biggest of all. Then if you have hot weather, you begin to not see that one, and these two would shrink. And then as it gets less and less pigment, you would not see that one, and this one would shrink. So that, in other words, the whole family seems to be genetically controlled, as we've indicated, but the last one to disappear would be the one normally present in largest amount. So if you had a really great technique that you could find infinitesimal traces, you'd probably find them going down in some ratio or rising in some ratio. So I still think my statement, at least as far as the available data would permit it, is so yet that the pink grape that is more intense would just have more cyanidin and not have malvidin, although I suppose it's possible it would begin to show a small amount of malvidin as it gets darker and darker. So in other words, in terms of the grape color, you have a concentration effect as well as a quality effect, but at least the quality effect, with cyanidin being more apt to show up under this condition, helps explain why you get dilute pink grapes and not dilute blue grapes. If delphinidin was the common product, then you would expect to get a pale blue grape. And if you find one, hang on to it, because it might have some at least nuisance value within salability of grapes. While we're digressing, I might tell you one little interesting story. There is a grape that I'm fascinated with. It's called Bonnet de Retard. I don't know why such a complicated name. And I personally got it in a vest pocket from South Africa as a small bud cutting legally into this country. It is under quarantine now and should be released shortly. The thing that's fascinating about it is that it looks like a little watermelon. It's got it's a sort of a reddish grape, about the color of a cardinal, I guess, and has stripes on it like a watermelon that are much less colored, whitish stripes on a red grape. And it seemed to me that'd be worth something as a curiosity, if nothing else. Why I was particularly interested in it, I wanted to study why these stripes didn't have the pigment and these did. I found very quickly with a microscopic examination that it was wrong. It's the same pigment and about the same concentration. What's changed is the surface of the skin. The skin's rough in this area and has more wax on it, so it appears white, just due to the, or whiter at least, due to the wax, whereas this is shiny, and if you rub off the bloom, it's like the average shiny grape. So if you polish one of these, the stripes remain, but the color then, the roughness still shows the stripe, but the color is the same. So I thought I had a cute example of a chimera or a local mutation within the tissue where it would be nice to study the formation of the pigments. It turns out that it might be nice to study other things. For instance, if you let one of these grape berries dry, it looks like so in cross-section. In other words, if you make raisins out of them, the water evaporates much more rapidly through the rough skin. So now all we have to do is cross that character into some Thompson seedless, and we'd have some raisins that would dry in 2-3 days probably. But that's another problem. I'll leave that to probably Professor Olmo's successor. It didn't do the job I wanted, but it's an interesting sidelight, I think. I'm not sure. I did say that all the wild grapes, but maybe 1 or 2 South American exceptions, are red, and even the South American exceptions have red as the predominant color. And the white condition seems to be recessive in all cases. And this is one reason why, again, the French hybrids and the ones more similar to the wild type tend usually, there are more examples that are red than otherwise. There are some relationships to the plant as a whole. After all, if the berry is going to synthesize anthocyanins, then the plant has to have the capability. And at least I don't think this works in vinifera. For instance, you may know that white Riesling, one of the identifying characteristics is the young canes are red, so clearly it doesn't always work. But in American species and French hybrids, apparently you can go by in the spring and drive by in a car if you want to and pick out all the red grape producers and all the white grape producers, because the first leaves in the spring are red on the ones that are going to eventually have red fruit, and white on the ones that are not. At least there is a publication that says this, and said they've only found 1 exception out of about 500 examples that they took a look at. So the general property and the physiological difference we may have a tendency to forget is throughout the plant. And even though we don't see the anthocyanin in certain other tissues, clearly the genetics make it possible, and under some circumstances we may find anthocyanins in other tissues or other phenols that are related to the ability to make anthocyanins. Well, I think we've belabored that point sufficiently. Let's that group of points. Let's talk about the flavonols and flavones. Quercetin, contrary to the anthocyanins, is found free, or at least it's so readily free that it occurs in wines at a very early stage and in extracts from the tissue, so that either it is present naturally free, or it comes, the glycoside comes off so readily that it appears to be free. It also, however, occurs as the 3-glucoside and the 3-glucuranoside. No other phenol in grapes has been found to be a glucuronic acid glycoside, except this one. This illustrates the fact that the more usual phenols have been reported, such as quercitrin or rutin, but it turns out they're probably not there. It's misidentified because they are glycosides of quercetin, but not the stated ones, because the rhamnose apparently doesn't occur in the grape, even though, as we've said earlier, the Rhamnaceae is a most adjacent family to the Vitaceae. While we're on that point, there are other reasons. I think we mentioned that the Rhamnaceae contain anthraquinone laxatives that we're glad aren't in grapes, but also they're exotic flavonols such as rhamnetin or rhamnetin, which is present in the Rhamnaceae. It's a 7-methoxyquercetin and is not found in grapes. So again, it seems significant and probably dietarily very nice that the grapes seem again to have just the most common types that one would expect. In addition to quercetin, camphorol, which is the B-ring monohydroxy derivative, and miracetin, which is the B-ring paragallo analog, are found, and there are the ratios as indicated in your notes, so that red grapes, at least in tests by Pascal Ribeiro-Guyon, had about 5% camphorol glucoside, 50% quercetin glucoside, 35% quercetin glucuronoside, and 15% miracetin glucoside. So you see the total would be about 85% quercetin derivative, and that would be a very high percentage. The white grapes had more of the camphorol and none of the miracetin. So again, there is this suggestion, which I think I mentioned earlier, that if you don't make the red pigments, one of the losses seems to be the ability to make the trihydroxy B ring. And that again comes back to our suggestion here. If you're going to lose the ability to make red pigments completely, you seem to lose both the anthocyanin formation ability and the trihydroxy B ring. And that fits nicely with our theory that, at least in the investigated cases, it's a fact that the cyanidin seems to be produced and not delphinidin derivatives. So it begins to hang together, and we think it's a fact, although certainly very few white grapes were looked at, and probably some more examples ought to be done. The low flavonol content of wine is partly because when the glycosides are removed, and they apparently do come off rather readily from the flavonols, the product is not very soluble. However, another reason is that it's been shown that the juice doesn't contain very much flavonoid. It had been shown in this case anyhow, and by our own work in general, that flavonoids are very low in non-teinturier juice grapes. So that the phenols of free-run clarified juice are very nearly exclusively, whether it's red grapes or white, non-flavonoid materials like caffeic derivatives, caffeoyl tartrate, if that's it, or more probably chlorogenic acid. The amount in skin fermented wines, red wines, would range from around 40 to 100 milligrams per liter of total flavonol. The flavones have not been found in fruit, and I don't think they will be. It's rather characteristic that they don't occur in fruit, but do occur in the floral, in the leafy and or bark parts of the plant. So they are not found in fruit yet, and probably won't be, and even if they are, they're not generally very soluble, so they probably would be removed during fermentation and not appear in the wine. However, they have been found in Vitis cinerea, but not, or either absent or very low levels in Vitis vinifera. And since Vitis cinerea is rather resistant, has a complex pattern of resistance to nematodes, phylloxera, and a few other things, they were thinking maybe this is one reason, so that they were trying to use the content of flavones, and not only flavones, but they were the C-glycosides of the non-oxygen link, but the carbon link glycosides in the leaves. So they were trying to use this in Germany in some studies to see if this is tied into the resistance. And it did not seem to be. It seemed to be a pretty good indicator, and the presence of these compounds was genetically dominant when cinerea was crossed with vinifera, but it didn't seem to parallel resistance. So it's an incidental indicator rather than a cause and effect indicator, apparently. The flavanones, the only one found is ampelopsin, and notice the name. Ampelography, you know, is the study of grape varieties and their interrelationships, and Ampelopsis is a genus within the Vitaceae. So in Ampelopsis, a species, Ampelopsin, was found. It is the flavanonoal equivalent to miracetin, that is the trihydroxy B ring. But it's not yet been found in Vitis, and if so, it'll probably occur in the wood, and we'd certainly expect to find taxipholin in grape wood, since this is the usual situation if you find quercetin in the leaves, and we do in high amount in the grape. It hasn't been looked for, but it's probably there. At least I'm not aware of anybody looking for it. Flavanones appear to be absent, and the low content of flavones is consistent with the woody condition as opposed to the herbaceous, as we've indicated earlier. Now this brings us to the amounts and distribution, and we started talking about that at the end of the period last time. If we exhaustively extract the whole grape berry, then we would have up to around 7,400 milligrams per kilogram of phenol in the fresh fruit. And these examples that I showed you last time are more typical, where the amount total would be a bit less than that. The typical value around maybe 6,000 for the red, or a little less, and about 3,500 or so for the white. The stems would contribute perhaps another 1,000 milligrams per liter or per kilogram if they were included. In wine, we would generally remove the stems, and if we had the same amount, that if all the phenol of the berry appeared in the wine, it had averaged about 4,600 milligrams per liter. If we compare these contents with other fruit, it's not particularly outstanding, and there are examples in your notes. The strawberry runs around 2,000, the pear around 4,000, and some of the pears with high tannin that are used to make perry, the cider-like drink from pear, are much higher than that. Peach around 2,000, apple around 1,700, and so on. So that, and the banana is pretty high, persimmon is outstanding, so that this content of the grape is not so unusual, particularly if you figure that this total is including the seeds. Now, in most of these other fruits, either there isn't much tannin in the seeds or phenol in the seeds, or we don't eat the seed. So in the case of eating an apple, you'd ordinarily peel it and not eat the core, whereas with a grape, you'd ordinarily put the whole thing in your mouth, and if you're as lazy as I am, you'll swallow the seeds, and generally you'll inadvertently chew up a few, so that you end up ingesting the whole phenolic content, whereas you wouldn't with most other fruits. Of course, the strawberry, the same thing would apply, and so on. Banana, you don't eat the peel, but you eat all the flesh, and you get a great deal of phenol that way. The seeds would represent around 2/3 of the total, as you can see from this figure. So if you were going to consume only the juice and the pulp, say, or even the pulp, in this case means the pulp that stays inside a cheesecloth, so you wouldn't ordinarily be consuming that, and you would be getting a very low content, and as we already said, we've shown that that's essentially non-flavonoid in both red and white grapes. And they're basically similar in content, basically identical. So that the seed would generally have around 2/3 of the total. And interestingly enough, we don't have enough examples to be too clear-cut on this, since Thompson seedless is our main one, and Corinth and a few others are possible. But generally speaking, if the grape lacks seeds, not only does it lack about 2/3 of the total, but what's left seems to be low. In other words, Thompson is rather low in total phenol content, even if one looks at the skin and pulp and juice content, so that there seems to be a vague tie-in with the production in the plant as related to where else it occurs. And I might digress at this point then and say another piece of information that our work has shown. It has been, well, I don't need to show it, I'll show it in just a minute. It has been said for a long time that the phenol content of grapes decreases as the grapes ripen. Well, that's true if you're talking about a concentration basis, but we were quite interested, does the berry synthesize phenol as it ripens? And it turns out that it does. If we take a look here at about, see, this is plotted as days from 18 degrees Brix, so that the 0 would be at 18 degrees Brix. And this is a combined data from quite a few different varieties with the polyphenols of one berry. And you see that the phenolic content per berry would increase up to within about a month of the ripe fruit, and from there on it's either nearly constant or maybe slightly decreasing, although there is a suggestion of a further increase in the later stages, even after 18 Brix. The main point being that if you look at the concentration only, the fruit is enlarging faster than it's making phenol. So the apparent idea is that you're not making any phenol, but it turns out you are. You're just making it more slowly than the increased volume of the fruit, so that there is a net synthesis per berry up until quite late in the ripening. On the other hand, and this is a more complicated graph, if we take a look at concentration, so phenol per gram is this black line, and we get a figure that looks something like that. Now that's a single variety, I think it was Semillon, and the kind of data that we put together for 8 or 9 varieties for that, well, 2 or 3 seasons, in fact, this being one of them. And this rather funny jog, then a rise, and then maybe a jog and a fall off here seems to be rather typical. And these jogs in the line don't seem to be artifacts. Now, maybe that particular one is, but certainly this kind is not. In fact, we've gone to the trouble of taking intermediate samples and find that it follows a curve. In other words, this variation is real, and it's not due to sampling error or analytical error or so on. And we have made some effort to tie it into weather, and it does seem to tie rather nicely to weather, in that a high phenol content seems to follow about a week after a period of cold weather. Now, unfortunately, these are vineyard studies, and cold weather in our vineyards, it means cloudy, and in the late summer anyway, and in a couple of cases meant a little bit of rain too, even though that's rare here. So that we can't really say whether it was due to the temperature or the light or the rain, but in any case we can say that the complex that gave cooler vineyard weather was followed by a rise in total phenol. Also, there seemed to be some evidence that there was certainly a different lag in the seed and in the skin. There was some inference that there was a translocation between the two. In other words, the skin would respond rather quickly, and the seed would appear to go oppositely. So and we could get as much as a 40% change in a week's time. So if you have a 40% change in a week's time related to weather, it seems hard to visualize synthesizing all that phenol. And so while we've said, generally speaking, phenols are in the cells where you find them, perhaps in the rather limited translocation space of the grape, there is some partial breakdown and then resynthesis. Another aspect, of course, is we're talking about extractable phenols, and in the seeds at least, with the highly lignified tissue, a 40% change may indicate a response of a solubilization type. Again, a very interesting area that we'd like to do more work on, but it hasn't had high priority and we haven't yet done it. It may be, and we think there is evidence, that the phenol content is related to surviving cold weather. In other words, the response to cold helps prevent freezing injury and this kind of thing, even though the cold in this case is nowhere near freezing level. The other things on here, pH change and so on, just show the correlations and show that most of these changes are pretty uniform and not very susceptible to the kind of weather change that produce these rather sudden changes in the phenol. So we were saying then that the typical grape contains a certain amount. What does the typical wine contain? A typical wine, if all the phenols of the berry went into the wine, should have 2,500 to about 7,500 milligrams of gallic acid equivalent per liter. And this would average out to about the 4,600. And that's not counting stems. If we added stems, well, we could get from 90, some stems are very low apparently, to 1,700 milligrams more. So that this would be high. Now, we've seen wines, we've made Calzin wines as high as 6,000 milligrams per liter, and I'll guarantee you, you won't drink much of that. It'll pucker you up worse than a persimmon, but it's possible. On the other hand, the typical red wine for commerce would be of the order of 1,200 to 2,000, and 2,000 would need age or other treatment. It'd be too strong. If you're going to sell it, it ought to be around 1,100, 1,200. The stronger whites and the pink wines would be around 500 to 800, and the whites would be under 300 milligrams per liter. And this is, of course, related to extraction of the phenols, particularly flavonoids, from the grape skin. There are wines made in Russia, the Caucasia wines, that still only have generally under 3,000 for the whites and under 5,000 for the red, and these are made, or at least traditionally have been made, by letting the skins, pumice and all, sit in the wine as long as you wanted, 3 years or something of that sort. So even in this case, where you've given every opportunity for the extraction, you generally don't reach the level that you would expect to reach. And one reason you don't is not that the phenol isn't getting into wine. I'm running out of time. The phenol does get into wine. I'll probably want to come back to this next time. But this is a chart showing the extraction from seeds. And you can see that with, say, 11% alcohol, for 24 hours even, you're getting up to 40 or 50% of the total phenol from the grape's seed. So that it isn't that it's held in the seed and can't get out. It is that it's precipitated by the yeast and the conditions of the fermentation. So the reason the wines are lower is not because it doesn't get in there, just because it doesn't stay there. And so I'll stop at this point. Next time.